Hi, I'm Huaming Sun, a PhD student in Shell Lab at Worcester Polytechnic Institute. In this video, I will talk about my poster using machine learning to understand the messenger RNA degradation mechanism in mycobacteria. The reason we study mycobacteria is because one of the mycobacteria, Mycobacteria tuberculosis, is the causative agent of tuberculosis, which remains a global public health crisis in 2020. MTB inhabits a complex and stressful host environment. It encounters antibiotics within the context of multiple stressors. Still, it's able to survive and proliferate. As a result, it will take over 6 months to treat TB. During this process, further developed antibiotic tolerance will make the treatment even harder and facilitate drug resistance. All of this raises a question: what makes MTB so successful in response to stress? Together with transcription, messenger RNA degradation determines the messenger RNA abundance. By altering messenger RNA abundance, messenger RNA degradation could contribute to stress responses through 2 levels of regulation. One is transcript-specific regulation, which could result in altered expression profiles. Another is global regulation, which could contribute to metabolic adaptation to energy-limited environments. Given such useful rule in stress response, there's still little we know about what controls messenger RNA stability. Previous research only finds limited correlation between individual messenger RNA feature with messenger RNA stability in MTB. However, the problem is that those features cannot explain the entire variance in messenger RNA stability. Similar analysis using linear regression in other bacteria also faces the same issue. In mycobacteria, we are specifically interested in leadered and leaderless genes, defined by if they have a 5' UTR, which could potentially affect messenger RNA degradation through translation efficiency and degradation initiation. However, the specific mechanism is still not clear. Altogether, a better understanding of messenger RNA stability through a more comprehensive analysis is very much needed and can be useful for TB treatment. To do that, we use machine learning to help us understand the mechanism by identifying the features that determine messenger RNA stability. After using drug to block transcription initiation, we collect RNA over time for RNA-seq to quantify messenger RNA degradation transcriptome-wide using non-pathogenic mycobacteria mSMAC. After calculating half-life through the degradation profiles, we split genes into 4 classes by their half-life. Then we collect messenger RNA features from 5 different categories. Each one of them have features carrying information about different parts of the messenger RNA. Random forest model will be trained to learn the association between features and the half-life classes, and will be tested by making prediction of degradation class. At the end, the model will rank the features by their ability to contribute to the prediction. Those important features have more distinct distribution over half-life classes compared to those that are less important. First, our degradation profile showed that not only messenger RNA are globally stabilized in response to hypoxia stress, but also such stabilizations are at different extent. Our analysis also suggests that the regulation of messenger RNA stability is achieved through a nonlinear and non-additive combination of multiple features. Features from different categories could be important to determine messenger RNA stability. As for leadered and leaderless genes, we find that some features are more important to predict half-life classes for leader genes, while different features are more important for leaderless genes. Such results indicate the degradation mechanism could also vary by genes. By looking at feature distributions over half-life classes, we can further explore how the feature value changes and the half-life changes. As you can see, the determinants could work differently to control messenger RNA stability. Further quantifying the feature importance suggests that the discrepancy of important features are not only existing at gene level, but also at the half-life class level. Each half-life class may have its unique features that are associated with messenger RNA half-life. Similarly, we are able to develop models to predict the extent of messenger RNA stability upon transition to hypoxia condition. Here, our results suggest that 5' UTR-related features are more important to predict such stabilization process than to predict degradation in log phase. Overall, we are able to develop machine learning model to predict messenger RNA degradation rate and the extent of messenger RNA stabilization upon transition to hypoxia condition, which could further provide insights on how mycobacteria survive stressful environment through a refined understanding of the complex regulation mechanism of messenger RNA stability. Thank you for your attention.